Hello everyone. So the paper I'll be discussing is called Sea Anemone Membrane Attack Complex Perforin Superfamily demonstrates an evolutionary transitional state between venomous and developmental functions. So the broad picture here is that gene duplication is a major driving force for evolutionary innovation on the molecular scale. And so here are some key terms. So recruitment is the generation of new animal toxins via duplication of physiological protein encoding genes. So this has been experimentally verified and observed in real life, where previous physiological, maybe developmental protein encoding gene, after a round or two rounds of gene duplication, it will gain a new function and may function as new animal toxins and be venomous for other animals. And reverse recruitment is actually the opposite of this process, where it's uh, a venom protein transforming back to a physiological protein encoding gene via gene duplication, and it is yet to be proven experimentally before the study. So the study focuses on members of Cnidaria, which are venomous and employ specialized organelles called nematocysts, and it specifically focus on members of the membrane attack complex and perforin family because it is able to lyse cells, and this function is very important for both venom and for producing venom and helping in development because it could get rid of the old cells. And so the two hypotheses that the research came up with is that reverse recruitment of venom proteins is able to occur, and that it can be transformed into endomesodermal cells because after a round of gene duplication. And also transitional forms between venom protein and a non-venom protein also exist, where the gene is expressed in both type of cells, the staining cells and the endomesodermal cells, and also it functions both as a venom protein and a non-venom protein. So on the right, we have an example of these nematocysts, which act as stinging cells to attack other animals. And there are quite a lot of information, but basically, they identified seven MAC members, 1 through 7, and they primarily focus on MACs 1 through 4, where 2 is exclusively found in nematocysts, 1 exclusively found in endomesodermal cells, and 3 and 4 are found both in um, nematocysts and endomesodermal cells. So on the trigger to the on the top, you can see that this is the relative expression level of these MACs 1 through 4, and the x-axis is the developmental stage of these animals. So as you can see, MAC2 almost retained equal amount of expression throughout the animal's lifespan, whereas 1, 3, and 4 had a peak in the developmental stage, and then its expression decreased when it reached adulthood. So this suggests that MAC2 plays a role for venom proteins because it is needed throughout its lifetime, while MACs 1, 3, and 4 probably has a role in developmental functions because their expressions decrease and it's no longer needed when it reaches adulthood. So also MACs 3 and 4 probably exhibit intermediate expression because it is found in both types of cells, and that the functions probably overlaps in these cells also. And on the bottom figure, you see that there's evidence showing that when you deplete these 3 endomesodermal MACs, the percent of primary polyps that reach adulthood actually decrease about 15%, which probably suggests that members of these MAC family are essential for proper nematostad development and their function in these developments. So this finding is significant because it unravels the evolution of a gene family of the MAC family, and that it's able to change from venom a non-venom protein to a venom protein, and then back to a non-venom protein after a few rounds of gene duplication, and it highlights the power of gene duplication to recruit these different proteins into different cells. And also, it shows that there are transitional states that exist where these proteins are expressed in both types of cells, and it functions probably both as a venom protein and as a non-venom protein. And this is exciting discovery because it is hard to piece together the exact evolutionary process that leads to change to genes getting new functions, but this is an example of that. And thank you for listening.